cells have any resting membrane potential?
No.
No. All right, so they have a graded potential. Is it a graded potential because we can change it by the amount of ligand chemicals that bind to channels or not? Yeah, we can make it steeper or we can make it more shallow. All right, so if we're using the sympathetic nervous system, then we have a steeper. Positive ions flow into the cell faster, takes less time. Or if we're using parasympathetic, we can have it take longer. Okay? So PS for parasympathetic, S for sympathetic effects. So that's the graded potential. Now, what types of channels? Could I erase this list over here on the board? You guys have that? And may I erase these three types here? You have it from Friday, I know. Okay, leaving this up here, I want to redraw the standard. So what types of channels are we talking about that affect our graded potential? As the cell is repolarizing, as it reaches a minus 50 to a minus 55, that hyperpolarization opens a particular type of channel. Do you guys remember? The funny channel. That's just such a weird name. Not very scientific. So funny channels open. Primarily sodium enters the cell, though some potassium will leave through the funny channel. And then what happens just prior to reaching the threshold of our action potential?
Transient.
The transient. So this is actually another threshold. Okay? It's a threshold for our transient T calcium channel. So that allows another flood of positive ions to enter into the cell. Okay? Both of those together, the funny channels and the transient calcium channels, bring the cell to this point. And what do we call that? Threshold potential. Okay. What type of channels open to create our depolarization? Because this is our action potential.
Long lasting?
Yes, the long-lasting what?
Calcium voltage gain.
Calcium. So in our other two examples, the ones that I erased, the skeletal muscle and the cardiac contractile, that depolarization leg of action potential for both are voltage sodium. Okay? So this is our big difference, the exception to the rule. So this is going to be long-lasting, voltage-gated calcium channel. Will open, yes.
That happens at the threshold potential? Or
The threshold potential is what opens those. Okay? That's what is the voltage-gated. This is also known as fast calcium channels, as opposed to the slow that we'll see when we get to cardiac contractile cells. All right? So that's this depolarization arm here. And then that depolarization opens our voltage-gated potassium. Okay? And And it's the slowly opening, slowly closing potassium channels here also that contributes to that depolarization. So voltage-gated potassium are closing, thus we're slowing down our loss of positive ions. And then at this point, we open the transient calcium. So that's what this potassium is referring to here. Okay? So while they're still open, we're slowly losing potassium. And then they eventually close. Sodium is gained. Calcium is gained through transient channels. Then the long-lasting fast channels open, and we see our spike with the voltage. So that's the standard. Remember, that's going to be about 100 times a minute if all we're looking at is the autorhythmic cells of the sinoatrial node. But that's not our average heart rate. So is it sympathetic or parasympathetic that comes to play as we slow the heart down to the average 70, 72 beats per minute? Parasympathetic. So let's look at what happens with that. I don't have a picture, but I have. So to slow it down, we do this by two things, okay? We affect, parasympathetic is green, right? So we affect the funny channels, and we affect the transient calcium channels through the parasympathetic nervous system. And there, well, I'll leave that up here, I'll put it up. So our neurotransmitter for, what's the nerve that's bringing the acetylcholine to the heart? Anybody remember what's our parasympathetic nerve to the heart? Vagus. Yeah, the cranial nerve, 10. So acetylcholine is going to bind to muscarinic receptors. And these are G protein receptors, all right? So two things can occur. It will activate an inhibitory G protein. By activate, I mean it's going to kick off the GDP and have a GTP added to it. But the effect of the inhibitory G protein is to inactivate adenylate cyclase. That's going to result in a decrease of cyclic AMP, and that's going to result in a decrease in the number of open funding channels. Okay? So they're not going to open until the current becomes much more negative. So that's one thing that it does, okay? That's this one right here. Those are the, well, it's the calcium one, so it also works on the funny channels. And then the other thing it does is it activates a stimulatory G protein, which binds to and opens potassium channels. What's that going to do to the membrane potential? It's going to make it more hyperpolarized. It's going to take longer. In other words, it's like trying to fill up a bathtub, the water being the positive ions of depolarization, by turning on the faucet. That's what's going on here. But then with acetylcholine, we pull the plug. So at the same time we're trying to add more positive ions, we're losing positive ions via the potassium channels. And so it takes longer to reach threshold. Okay? We start at a lower level and it takes us longer to get to threshold because we're losing ions at the same time we're gaining them. So what that does is it increases the time between action potentials, makes fewer action potentials per minute. We require those to trigger the ventricles to contract so we have a fewer heartbeats per minute, a slower heart rate. Okay, does that make sense? If you understand the overall? So if we go back to our regular autorhythmic, what's triggering this is the funding current opens. Okay? And we add both positive sodium and calcium to reach threshold at 100 beats a minute. Acetylcholine comes along, and we have fewer funny channels. They don't open as early, so it takes longer to get to this point. Plus, we're losing positive ions through our potassium channels, so it takes even longer to get additional positive ions into the cell. Okay? That's how we slow the heart rate down to our standard 70 beats a minute if you're looking at the average. Now, the heart rate can be slowed even more by continued activation of the vagus nerve, all right, through a vagal stimulation, or, as we'll see when we do talk about cardiac output, by the force of the heart's contraction. So someone with a strong heart doesn't need to contract as often. For instance, average heart rate, 70 beats a minute, average stroke volume, that's the one volume for each ventricle when it contracts, about 70 mils. 70 times 70 brings you to about 5 liters, 4,900 mils. Okay? So average cardiac output per minute is about 5 liters of blood. You only have about 5 liters of blood, so obviously it's being pumped out over and over and over. Now, let's say that someone exercises, and with each heartbeat now, their heart is stronger because it's been exercised like any muscle. And instead of 70 mils, it can contract and pump out 100 mils. So what does a heartbeat have to be to pump out a 5,000 mils? It only has to be 50 beats a minute. All right? So we don't want to pump out too much. Our blood pressure would rise. So the body maintains a balance between its need for oxygen and blood pressure regulation. So the stronger the heart, the greater the volume per contraction, the fewer the number of contractions that need to occur. So that's why we see that balance. So we talked about a weak and thready heartbeat. So if there's not much blood volume, so whenever the heart contracts, it's just we've lost blood through hemorrhaging. So now when the heart contracts, it's only pumping out 30 mils instead of 70. All right, with a heart rate of 100, you still only have a cardiac output of 3,000. So the heart rate has to get increased to try to get the same amount of blood circulating every minute. So that's we'll look at that relationship when we look at that blood pressure. Okay, so slowing the heart rate. Now, we do the reverse when we increase the heart rate. So I'll leave that diagram up and erase the green structures here. So now sympathetic nervous system is going to increase heart rate. So we're looking at norepinephrine or epinephrine. Epinephrine comes primarily from the medulla. Norepinephrine would be from our sympathetic nerves via the thoracic spinal cord. It also is going to bind to a G protein. So we have G protein activation. This is going to activate adenylate cyclase. Therefore, that's going to increase the number of cyclic AMP, which will bind to our funny channels, as you can see here in the diagram. And they will open earlier. So this increases the number of funny channels that are open. And it also, they open earlier, meaning they open at a less negative, like a minus 45, instead of, they don't have to wait until they get to minus 55. So they're going to open, say, here, instead of down here. We also increase the rate at which we add positive ions because they will open the transient calcium channels. So these transient channels here, more of those will open. So not only do we not have to wait for the cell to hyperpolarize as much, but we have more positive calcium channels open, and we're adding calcium at a faster rate. Okay? So this will happen, and more transient calcium channels open. So it's kind of the reverse, except for the potassium component, but the calcium and funny channels will open earlier and more of them. I'm not sure. There will be as soon as this stuff gets uploaded onto the internet. All right, so what you have to do, and it's if you break it down to those two components, here's our standard view. Here's our 100 beats per minute, okay? And we slow it down, then we lose more positive ions via potassium, and we are hyperpolarized because our funny channels don't open. For sympathetic, our funny channels open earlier, and we gain positive ions via calcium. So the funny channels are affected in both. So parasympathetic is going to not allow as many funny channels to open and allow potassium to leave the cell. Sympathetic, funny channels open earlier and more calcium comes into the cell. So they both share funny channel effects, but acetylcholine affects potassium, whereas norepinephrine affects calcium channels. Okay, if that helps you keep it a little bit straighter in your mind.
Professor?
Yes?
They both hyperpolarize, but the sympathetic, it does not hyperpolarize as much.
Exactly. So in other words, the funny channels open because of the hyperpolarization. Let me diagram that. I kind of have it here, but let me do it. So sympathetic effect, let me draw some lines up here. So this is threshold potential, voltage gated, long calcium channels open.
Does that ever change?
No. Okay? Given a particular situation, if we change the other, we can change it if we change parameters around. So certain medications may change it, but typically no. This is going to be when our funny channels open.
What's TP?
Threshold potential. Okay? So on our normal activity, we depolarize, we reach hyperpolarization, and funny channels open. And then our transient channels open. Okay? And then we reach threshold and we see our action potential. So that's standard. Okay? Sympathetic effect. We don't have to reach as negative a level before funny channels open. Okay? Plus, our transient channels open earlier, so more calcium comes in. So it's not just that we do this earlier, we actually have a steeper gradient as well. So both of those effects then will allow us to reach our action potential earlier. Okay? And then for our parasympathetic effect, we hyperpolarize more because the funny channels aren't opening without cyclic AMP. We lose potassium, so it's taking longer to get to a positive point. So, that's almost the same. So our gradient becomes more shallow. Okay, so where both of these two things are happening, funny channels open earlier, more positive ions are added. Spotting channels open later, more positive ions are lost. So between those two, it takes longer or it takes less time to reach the threshold of the long-lasting calcium channels. Is that a little bit clearer? Okay, those two events are happening there.